This is the fourth video of the fluorescent and bioluminescent fusion proteins for neuroscience lecture. In this video, we will discuss the use of bioluminescent reporters, particularly the use of luciferase enzymes to study gene expression regulation. So for this type of application, we use luciferases, which are oxidative enzymes that catalyze a chemical reaction that produces bioluminescence. Bioluminescence is a type of chemiluminescence, which is simply the term for a chemical reaction where light is produced. And this is different from fluorescence, since a chemical reaction is involved in the production of light. So luciferases are commonly used as a tool to study gene expression at the transcriptional level. It can deliver from 10 to 1,000-fold higher sensitivity increase than fluorescent assays. Luciferases are also naturally found in several organisms. We can find them in fireflies, from where we get the firefly luciferase, or other animals such as the marine animal sea pansy, from where we get the Rhinella luciferase. And these enzymes differ in their substrate, cofactor requirements, and also the type of emitted light. And all these differences can be exploited in different applications that we will see in the next few slides. Basically, this system uses the fusion of the luciferase gene to the promoter of a gene of interest to study gene regulation. And these plasmids containing this construct are transfected into cells or inserted into animals to generate transgenic animals to study gene regulation. And whenever our gene of interest is expressed, we will detect an increase in emitted light by the luciferase enzyme. And if you want to know more about the luciferase applications, you can visit this website, where there are several applications where you can apply luciferase. As I just mentioned, different luciferases have different properties. For instance, firefly luciferase uses luciferin in the presence of oxygen, ATP, magnesium, and they produce light by different intermediates. They end up producing another intermediates plus light. On the other side, Rhinella luciferase requires only choloenterosyne as a substrate, and in the presence of oxygen, they also produce light. In addition, firefly luciferase produces a greenish yellow light around 550 and 570 nanometers range, while Rhinella produces a blue light at 488 nanometers. There are also other types of luciferases depending on the animal that we take it from. So these enzymes can be used in dual reporter assays due to their differences in substrate requirements and light output. So the dual luciferase reporter assay system provides an efficient means of performing dual reporter assays. In this dual luciferase reporter assay, the activities of firefly and Rhinella luciferases are measured sequentially from a single sample. The two luciferase genes are contained in different plasmids and regulated by different promoters. Firefly is regulated by a target promoter for which we want to study the regulation. And the Rhinella gene is regulated by a viral promoter that is low but constitutively expressed. These two plasmids are transfected into cells. And if your gene of interest is expressed under your experimental conditions, upon addition of the proper substrate, firefly luciferase will be transcribed and translated, and light will be produced in the presence of this appropriate substrate. The emission of light reflects the activity of the target promoter. Then this reaction can be stopped, and Renilla reaction can be initiated by the addition of the proper substrate. The signal coming from Renilla is just used as an internal control for transfection efficiency, or amount of cells present in the assay. And then the signal from Firefly is normalized respect to the Renilla signal. In this slide, you can see the use of bioluminescence for non-invasive imaging in vivo studies. For this, we need to generate a transgenic mouse that expresses luciferase under the control of a particular promoter of interest. At certain time points, animals are intraperitoneally injected with the luciferase substrates, and light is recorded from a ultrasensitive camera. Then the advantage of bioluminescent reporters compared to fluorescent reporters is that fluorescent reporters requires exogenous illumination to emit light. And this technique is not appropriate for long-term and quantitative imaging for several reasons. One of those reasons is that fluorescent reporter is bleached by repetitive illumination. Also, repetitive exogenous light illumination causes phototoxic damage to cells. And also, exogenous illumination perturbs the physiology in light-sensitive tissues such as retina. In contrast, this bioluminescent imaging using luciferase reporters does not need exogenous light illumination, and the luminescent reaction can be quantitative by using specific imaging and translation analysis. This also has extremely low background. So this imaging method is particularly useful for longitudinal and quantitative imaging. An example of in vivo bioluminescent imaging can be found in this paper, where the authors performed longitudinal analysis of neurogenesis during induced encephalomyelitis. For this, they generated a transgenic animal expressing the luciferase under the control of the double cortin promoter, which is a specific marker of neuronal progenitor cells. And then they monitor neurogenesis in living mice by using a pharmacological induction method for encephalomyelitis. After different days of drug exposure using this molecule, they acquire images and they acquire the luminescent intensity, and this one is measured as full induction of double cortin during induced encephalomyelitis, demonstrating the presence of neurogenesis during inflammatory processes. You can see here in the negative control under PBS conditions, there is no luminescent coming out, or an induction of the luminescent coming out. While in the treated mice, you can see an increased signal within the brain of these mice upon encephalitis induction. So, as usual, I leave you here with a short quiz that you can complete at home, and that to revise all the concepts that we just learned in this video. And we will also cover some of these questions in class.